This PowerPoint is going to cover cell respiration. This video specifically is going to cover glycolysis, the first step of cell respiration, which implies the full breakdown of food for energy, or chemical energy in food to a chemical energy that we can use in our cells, which is ATP. So a very common source of food is going to be sugars, or glucose specifically. And the formula for glucose is C6H12O6, meaning you have 6 carbons, 12 hydrogens, and 6 oxygens. The chemical formula is C6H12O6, or glucose, or sugar, plus oxygen to make CO2, H2O, and lastly ATP. So this reaction altogether is going to be an exergonic reaction, meaning you're breaking something down, something large and complex, into something small and less complex. But the formation of ATP is actually going to be endergonic because we are using energy in order to make it. So we're putting energy into that ATP. So as kind of a review, if you're a molecule that is gaining electrons, usually carried by hydrogen atoms, we call this reduction. So if you are gaining electrons, you are being reduced. If you're losing electrons, then you're therefore oxidized. So in this case, you have this large complex molecule with many carbon-carbon bonds. What helps form the carbon-carbon bonds in sugar is actually the electrons. If you remove electrons, you're breaking a bond somewhere within this molecule. And what we do is we break apart those carbon-carbon bonds, extract those electrons, and we give them to these things called electron carriers, which we'll cover in a second. Now, when you break those bonds apart, you yield the carbon and oxygen from it in the form of CO2, a smaller, less complex molecule. So an exergonic pathway or a catabolic process to break down this sugar. Now this oxygen here, this is actually receiving electrons at the very end of aerobic cellular respiration as the final electron acceptor. It receives electrons, therefore it becomes reduced. Then it picks up some hydrogens and forms H2O. And we'll get into the details of that later. So whenever one material is reduced, another is oxidized. So if we're looking at this formula here, your C6H12O6 goes from large to small. It goes from electron-rich to electron-poor. Therefore, it's being oxidized. So glucose is being oxidized into forming CO2. And then oxygen becomes reduced into forming H2O. Okay, so those electron carriers. So NADH is one of the electron carriers we'll be talking about. It actually carries 2 electrons. Same with FADH2. Technically, it carries 2 electrons, and those 2 electrons are actually dropped off into the electron transport chain. So NAD plus is not carrying electrons. You could say this is the electron carrier in its oxidized form, and FAD is the electron carrier in its oxidized form as well. So these are the electron carriers in their reduced forms. Here's the electron carriers in their oxidized forms. So 3 major metabolic processes are going to be used to break down glucose for energy. So the main things are going to be glycolysis, which is the very first step. So as soon as glucose enters the cell, glycolysis takes place to convert glucose, which is a 6-carbon molecule, into 2 3-carbon molecules. And then we have cell respiration, or you could say aerobic respiration. And that's the process of breaking down pyruvate into inorganic material, meaning it doesn't have hydrogen or oxygen simultaneously. And like I said, it's aerobic, so it's going to require oxygen. That's going to all take place within the mitochondria. So any aerobic process is going to involve your mitochondrion. Anything outside of the mitochondrion, when it involves breaking down sugars, for example, It's going to be an anaerobic process. So another way we can actually generate energy indirectly is through fermentation, which is going to happen in the cytoplasm. And that's where you actually take pyruvate, you convert it into other organic molecules depending on what type of organism you're dealing with, and it does not require oxygen. So like I said, we're going to talk about glycolysis in detail. So glycolysis is going to happen in the cytoplasm. So as soon as glucose enters the cell, glycolysis is going to occur. By the end of glycolysis, you're going to create 2 net ATP. You're going to create some charged electron carriers, or electron carriers that are reduced electron carriers. And then lastly, you're going to produce 2 organic 3-carbon sugars called pyruvate, or pyruvic acid. Now, like I mentioned earlier, glycolysis is it's an anaerobic process. It's happening in the cytoplasm, so it's not going to require oxygen. So it's anaerobic. And as mentioned earlier, glycolysis happens in the cytosol. So we're going to break down glycolysis into 2 stages. We're going to have the energy investment phase, so reactions requiring ATP. We invest some energy into breaking down glucose. And then we have the reactions that produce ATP. So after we've invested the ATP, we get our return on investment to produce more ATP. We net 2 ATP. So here we're going to summarize the investment phase. The end result is you're taking a 6-carbon glucose and you're splitting it into 2 3-carbon glyceraldehyde 3-phosphates, or G3Ps. And your investment is 2 ATP molecules. So phosphates from each ATP are going to be added, so you're phosphorylating that 6-carbon glucose on carbon number 6 and carbon number 1 of glucose. And what you're going to do there is form what we would call fructose 1,6-bisphosphate. So the bi here means 2, and the phosphate here means phosphates. So you have 2 phosphates on this molecule, fructose. And those phosphates are going to be on the carbon number 1 and carbon number 6. And then the enzyme aldolase splits the molecule into 2 3-carbon molecules, one of them being glyceraldehyde 3-phosphate. So to talk about the cash out of glycolysis, the reactions producing ATP. First you have glyceraldehyde 3-phosphate, which is phosphorylated without ATP as a donor, meaning you're not investing ATP anymore. You're taking an inorganic phosphate floating around in the cytoplasm, and you're bringing that and attaching it to glyceraldehyde 3-phosphate, giving it 2 phosphates. And in that process, what you're doing is you're also simultaneously harvesting electrons, plugging them into your electron carrier NADH. Or you're plugging it into NAD+, which then becomes NADH when it receives its electrons. And then you have more reactions which will yield 1 ATP per glyceraldehyde 3-phosphate molecule. So we call this part of the pathway substrate level phosphorylation. So this is a way of creating energy, creating ATP, without the use of ATP synthase, which might sound familiar from previous biology courses. We're creating ATP at the substrate level. We're not using the facilitated diffusion of hydrogen through a transport protein within the inner membrane of the mitochondria to create ATP. We're doing it using substrate and enzymes in the cytoplasm. And what you're phosphorylating is your ADP to make ATP. So the final product at the end of your investment and your cash out or your payout is 2 3-carbon molecules of pyruvate, some ATP, and NADH. So here we have the preparatory phase or the investment phase. So you've got glucose, that's 6-carbon glucose. You invest ATP. You expend that ATP, yielding ADP. Now that phosphate's attached to your glucose. Then you invest another ATP. So now this sugar will have 2 phosphates on it. Then what you're left with through another reaction, through another enzymatic reaction, aldolase, is you're left with glyceraldehyde 3-phosphate, which is important to move on. And then you have the dihydroxyacetone phosphate, which you convert into glyceraldehyde 3-phosphate. So you're left with 2 glyceraldehyde 3-phosphates. So now when you want to cash out, what you're going to do is you're going to take this enzyme, glyceraldehyde 3-phosphate dehydrogenase. What that's going to do is it's going to splice in inorganic phosphate into this molecule. And simultaneously, it's going to strip away electrons and supercharge your NAD or NAD+, forming NADH. And you're going to be doing this twice because remember, you have 2 glyceraldehyde 3-phosphates. So here you have your glyceraldehyde 3-phosphate, which is then converted into a molecule with 2 phosphates attached. So the second phosphate you attached is inorganic. It doesn't come from ATP. You used inorganic phosphate to do it. So it doesn't technically require energy from ATP. Then in this 7th reaction, you have a phosphoglycerate kinase. So as a fun fact, a kinase is actually a type of enzyme that phosphorylates things. So here what you do is you take some ADP and it's going to strip away those inorganic phosphates to make ATP. Or you could say it's just going to strip away phosphates from this molecule to make ATP. Then you go through a few other enzymatic processes that shuffle around atoms. And lastly, you end with pyruvate kinase. What pyruvate kinase is going to do is it's going to take away that final phosphate to form ATP. So you've invested 2 ATP at the beginning. You invested 2 ATP at the beginning per glucose molecule. And then you're giving yourself, or you're creating 4 ATP as a result. That's going to yield a net of 2 ATP. So here's the summary. So what we've talked about so far is glycolysis. So glucose enters the cell, glycolysis occurs, yielding pyruvate, yielding ATP, and yielding NADH. Now in the presence of oxygen, you're going to be sent to the matrix of the mitochondria and go down this way. If you're deprived of oxygen, or if you're not in the presence of oxygen, you're going to enter fermentation, which we'll talk about last.